Have you ever wondered how life would be without saliva? Patients with autoimmune diseases and patients receiving radiation therapy as a treatment for head and neck cancer can be left with very little saliva. This can be due to a healing response that does not fully resolve after injury and leaves behind a permanent scar known as fibrosis. Fibrosis interferes with salivary gland function and leads to low saliva levels. Currently, there are no treatments that target salivary gland fibrosis to restore saliva production. My thesis work utilizes a reversible fibrotic model to study how one cell population can contribute to fibrosis. Those cells are endothelial cells, which line the inside of your blood vessels. My main question is if endothelial cells contribute directly to fibrosis by undergoing a transition into myofibroblasts, which produce the scar tissue, or if they contribute indirectly by secreting factors that recruit other cell types, like immune cells and myofibroblasts, which then produce the scar tissue. To answer this question, I performed single-cell RNA sequencing on all the RNAs being produced by endothelial cells with and without injury. I identified a unique population of endothelial cells that have a gene signature consistent with a direct contribution to fibrosis by converting into myofibroblasts, which produce scar tissue. Interestingly, these same endothelial cells also have a gene signature consistent with an indirect contribution to fibrosis by recruiting and activating immune cells and myofibroblasts. My results suggest that endothelial cells can contribute both directly and indirectly to fibrosis. Currently, I'm performing experiments using a method that traces endothelial cells before and after injury to look for further evidence for a direct contribution to fibrosis. I'm also using organoids, which are mini organs grown in a dish, to study indirect contributions to fibrosis. My work will open up opportunities for endothelial cell-based therapies for autoimmune disease and cancer patients to limit fibrosis.